All right, let's get started. So our next speaker is Dr. Brian O'Callaghan. He's from EMSL. He's developing cutting-edge technology in EMSL. So his house title is Cellulose Hydrolysis Studied by Infrared Nanospectroscopy.
Thanks, Sehong. Okay. Yeah, thanks for the introduction. Thanks for attending the talk. Today, I'm going to talk about our work with Professor Yang to study cellulose enzymatic hydrolysis using infrared nanospectroscopy. So an overarching theme of our motivation of our work is to extract more usable energy and biofuels from cellulose. So cellulose is a natural abundant material and could be used as a good source of energy. However, getting usable fuels from this is difficult to currently, requires high enzyme yields, and it's just kind of a rate limiting factor there. And there's a lot of current research undergoing right now to get more efficient ways to get usable fuels from cellulose. So to kind of engineer better ways to get catalytic conversion into, for example, ethanol for a lot of good applications. And one of the difficulties here is that there's a hierarchy of length scales that kind of are at play in extracting these fuels. So the underlying glucose units are the sub-nanometer scale. However, it's intertwined in the mesoscale, micron scale cellulose fibers, which are interwoven and need to be unraveled to get to this underlying energy unit. So there's been a lot of work, electron imaging and electron diffraction, other scattering techniques to get at the structures of cellulase, and we've been able to get atomic scale images and structures of this protein. And that, combined with MD simulations, has given us a good model on how this protein functions. So the cellulose, the binding module, grabs onto the cellulose, and that kind of guides the motion along the strand. And the catalytic domain, that's what breaks this strong hydrogen bonding in the cellulose and creates these smaller glucose units. And that's what we kind of want to understand in order to get at to improve this catalytic process. So this cellulase is a natural method to extract simple sugars from cellulose, and is a good potential technique to get this usable biofuels from cellulose. It's natural, self-driving, but currently it's been difficult to control and to scale up. So there's a lot of limiting factors requiring high enzyme doses to get a usable amount of fuel, and the reaction rates are tail off pretty quickly. So we want to understand specifically from a nanoscale perspective, what's limiting this, what steps are undergoing here that could be improved upon, and how we can get the process. So we're specifically focusing on the exoglucanase, which again operates by the protein traverses along these cellulose strands, breaks off the smaller subunits here at cellobiose, which is 2 glucose units. And then there's other protein processes that convert it to glucose, but we're specifically focused on how this protein binds to the surface, what chemical reactions then subsequently occur, and on what scale. So the technique that I'm implementing is nanoscale infrared imaging and spectroscopy, otherwise known as AFM-IR. So it combines atomic force microscopy and infrared spectroscopy, which this provides chemical imaging through infrared vibrational spectroscopy, combined with the nanoscale spatial resolution afforded by atomic force microscopy. So the schematic kind of shows the technique that we're using. So the AFM tip, this scans across the surface and gives height profile of the sample morphology. What we then do is focus infrared light onto the sample underneath the AFM tip. And this creates, so the sample absorbs infrared light when matched with its vibrational frequency. This heats the sample and creates small thermal expansion. This thermal expansion is actually detectable with our AFM and is specific to the material chemical composition and allows mapping of vibrational features. So here's a protein layer, and we're mapping the amide 1 response specific to protein with nanoscale spatial resolution. And by tuning our infrared source, we can get a spectrum comparable to FTIR studies. This gives us a definitive confirmation of the chemical content of the sample. So here's protein. So we see amide 1 at 1650, amide 2 1560, and other peaks that we can assign to the specific protein. So a little bit more on the experimental setup and its capabilities. So we use a pulsed IR source, and this resonantly drives a bending mode of the AFM cantilever. So it's specifically the second bending mode, which then allows the simultaneous measurement of the infrared sample local absorption, as well as the sample morphology. This has been used to demonstrate down to single protein spectroscopic imaging here at surface with dispersed thioglobulin and ferritin. So the dimensions are consistent with a single protein, and it shows up as bright contrast in the IR absorption map. So this is amide 1, so 1650 specific to protein, and you can also do these point spectra, which gives us a little more detail on and confirmation that this is actually protein present. Also, it's, here's an example of being used to study organic matter in mineral surface, so specifically oil shale, and the peak at 2920. It's a bending mode of OH, and it's indicative of organics, and again can be used to map this at the organics at nanoscale. This also has been very successful at imaging polymers and nanoplastics. So here's an example of a block copolymer, which is two different polymers joined together at the nanoscale, and we're able to image and identify what polymer is present. 25 nanometer spatial resolution. So the experimental setup we use for this, so we have a abacyl cellulose deposited on a silicon surface. We then expose it to different cellulase, so CBH1 and EG cellulase for 18 hours. We do our infrared nanoimaging before and after the reaction to probe chemical changes. Here's an AFM image of the cellulose after reaction. So the IR spectra average over this region, they're shown in this graph here, and so black is before and red is after, and there's significant spectral changes that have been that appear after reaction. So to get a little bit more detail on what changes occurred, we do take a subtraction of those two. So that's the black line there. That's the differential absorption after minus before. We see peak at 1650, 1540. That's consistent with the CBH1 peaks, which indicates that there's protein absorbed to this cellulose. And we also see decrease in peaks at 1040 and about 1180. And these are consistent with CH or CO stretch, and the decrease in those two peaks correlated with the protein absorption, and a weak increase in the C double bond. So this is giving us some evidence that there's actual chemical change to this cellulose upon reaction. And we can do this at the spatially resolved across this strand here. What I've done here is we've captured an array of points spectra across this strand here. And the spectra are stacked in the y-axis. So the x-axis is the frequency axis, and we do this across about a micron section of this cellulose. Red is increase in absorption, and then the darker color, the cooler colors are reduction in the higher absorption. Amide 1 and amide 2 are present, and you can see a little bit of its heterogeneity along this cellulose. So it's indicative of protein adsorption. And we also see a decrease in the CO and OH band. You can start to see any kind of correlation between protein presence and chemical change. Looking at the chemical structure of cellulose, we're kind of looking at the different interpretation of these spectral signatures, but the potential idea is that we're splitting these bonds between the glucose units, creating a CO double bond. So what this can give us is actually a hyperspectral data set. So we have images at each spectral frequency, and this can be stacked in this 3D array. So at 1650, that's our protein. So we see that there's higher a positive contribution. So this is the positive protein, the increase in protein absorption at 1650, and there's a little bit of heterogeneity there. And then there's also the decrease in the CO and the CH, the CO and the OH. So that's about these blue slices here. And what we can further do is to do a covariance calculation. So this is giving us correlation between the presence of one peak versus the presence or absence of another peak. And this is shown in the center graph. So cooler colors are anti-correlation. So one peak increases and the other one decreases, and the redder colors are the opposite. And so what we see is, especially if we look at different slices of this graph, so 1670, that's the amide 1, the presence of protein has reduced correlation with and negative correlation with the CO and the OH. So this is further confirming that, yeah, it's the protein that's associated with this chemical change in the cellulose. And this is spatially correlated, so this is the same. This is more of a direct measure of chemical changes associated with protein presence, which can't be done with any other technique, the nanoscale resolution. So a bit of an outlook on what we want to do next. So this was all that work was done ex situ. So kind of just different timestamps. But what we really want to do is to try to do this real time in liquid. So this could rely on a technique that developed here in EMSL. So it's a way to do in situ chemical nanoimaging based on an in liquid AFM, and we illuminate this AFM tip from below through an ATR IR transparent prism. And this then allows us to interrogate our samples in liquid and to get the same kind of chemical nanoimaging, but done with the protein in motion. I'm in real physiological conditions. And the geometry gets around the strong water absorption and a lot of background contributions by illuminating from below, and can get us a way to measure this with high resolution and in liquid. So a bit of proof of concept of this technique we've done. This imaging of this crystallized catalase structure in liquid. This is a protein layer that's about 150 nanometers high. And we can park at one point and obtain an IR spectrum of this. And we see that the IR spectrum is comprised of three main conformational contributions. And by fitting to the sum of three Gaussians, we can assign the different relative contributions between beta sheet, alpha helix, and random coil. This is doing protein conformational analysis in situ. And we can also do this spatially resolved. Pointers out, but we can do this with about 100 nanometer resolution. So yeah, in conclusion, so I did this IR nanoimaging of this enzymatic process of cellulose. So what our differential absorption map shows that there's distribution of bound protein, and we can do this correlation analysis to show that the protein is associated with chemical changes in the cellulose, specifically decrease in CO and increase in C below. And this can be done spatially resolved with tens of nanometer spatial resolution. And yeah, thank you for your attention. This is the team I work with, and happy to take questions.
All right, we have time for a question or two.
Very interesting work. Could you comment on the peak shapes that you get with this method as compared to conventional infrared microscopy? So in conventional infrared microscopy, you oftentimes you see changes in the shape of the peak due to me-scattering and other things. And are you immune to this, or how do you relate the spectra that you get back to whatever is known from the literature?
So there's a direct correlation to IR absorption. We're kind of immune to scattering effects in that way. So it's directly related to the imaginary part of the refraction. Yeah, there's not a lot of interpretation, fortunately.
All right, any other questions? Okay, let's thank our speaker one more time. We have one more virtual speaker up next.
Another laser. I guess the virtual.
Okay, so our next speaker will be Professor Vidya Susila from Clemson University. Her title is Fine Rules Chemical Plasticity in response of nutrient heterogeneity impact roots decomposition.
Can you see my screen? I think I have shared my screen. Oh, shoot.
Yeah, I think you might want to swap the view.
Yes, yes, sorry. Stop share.
There you go. We can see now, Vidya. Thank you.
Okay, so